Thank you, Federica, for the kind presentation. And Danilo, as I was introduced, I'm the head for the Genomic and Epigenomic lab. And my goal today is just to give you an overview of what we do in, not in the shadow, but in our lab of Genomic and Epigenomic. Let's try that we try to do our best to be a tool for the also for the new PRP project that you have heard during all this seminar. And by the way, thank you to be the closing section for these things. So just a few slides of introduction. We are a facility dedicated to RNA and DNA sequencing. But not only, we also still use a lot of genotyping with arrays, especially for the part of epigenomics. Some of you might well know that, but among this activity, I want to point out that we are a proper service. Proper certified provider. What does it mean? It means that we have been certified by an external advisor, actually from the company, that we do produce trustable data. The process was pretty easy. You just need to prove that you process hundreds of samples, and then they send you a blind sample to analyze. You ship back the data, you ship back your analysis, and according to your claiming, you get this label that indeed, whenever you use this kind of platform with this kind of certification, you will obtain the same kind of results. And we are among the 50 or more lab in worldwide that do this kind of things. I'll try to jump as much as possible the techniques, also because you have seen many of them during the previous seminars of people in my lab. Just to give you an idea how we ends up building this kind of lab. First of all, we haven't built from scratches. We have built it from an existing lab. This was a really clever idea because we improved on the experience that you already gained on the field. We are working on the field since 2004. And when we decided to do the first upgrade of the lab, we just looked to the forecast of what was available in the field. And as you see, we decided to go for the top technology in the field. That is actually, in our case, the Illumina sequencer, but also to include the technology that were not so well present in the market, but definitely disrupting in the sense of what kind of data you can achieve. And we have done not only in the term of data production, but also in the term of what we can really produce in the lab, also including automatization. But this may be a talk for another seminar. Just to give you a quick fresh up of how the next generation sequencing work. It doesn't matter which is the technique. You always have to go through these 3 steps: library prep, cluster generation, and sequencing. But what is really changing is the amount of data that you can gain from the same effort that you put in the lab. So why build such a kind of core facility? Simply because you producing more data, it means also that you can lower the price for the single researcher on the sequence that you want to achieve. And it's always a matter of balance between what's the real question that you have behind and why you are using this kind of tool. It's a really nice tool, but it's still a tool. It's not giving you anything particular. And since we are also interested in the epigenomics, I want to point out that, for example, we, as anticipated, we do a lot of epigenetic. We talked about epigenetics in previous session, and among the many ways how epigenetic can act, there's definitely the methylation of the cytosine. Actually, this is one of the many ways how you can achieve the epigenetic effort, but this is seen from the point of view of what you can reveal. And how you reveal it, it's simply because you, when there's a methylation in the cytosine, I don't know if there's the yes, there's the slides. If you have a cytosine that is methylated, a process that is called bisulfite conversion is not happening, while when it's not methylated, you have a change in the cytosine to another base, and it's so this process is basically what is going to happen exactly like when you want to detect a mutation, and we well know through the sequencing how to detect a mutation. So it's an indirect way to assess the state of methylation. There's many limitation in this kind of process, but still this is done not through sequencing, is done mainly using the arrays. And the reason why it's done using the arrays because it's been the gold standard for many years. And it can be easily scaled up. We process hundreds of samples per week in this way. And it's definitely robust in a sense, but just to give you an example, this is a tool, but the application can be of many different fields. I won't go too deep in this part because it's out of the PRP project, but we have done a lot for chromosome instability, but also for cancer topic, and the specific case of our collaboration with Neuromet, also in the population studies, where we analyze in this way more than 5,000 samples, still counting, because the overall population that they have is about 23,000. You can achieve the same things through the sequencing. For example, you can achieve the same things using the reduced representation bisulfite sequencing. And we have done, for example, in genomic analysis of family with specific mutation. The difference in this case is obviously that you are not detecting through a specific probe, but you're detecting through the sequencing. Still, is indeed more robust, but there's a lots of difference in this. And as I said, the devil is in the detail, because if you have a look in the left, you have what you need for the arrays. In the right is what you need for the reduced representation bisulfite sequencing. As you can see, there's a striking difference on the starting amount of material. To give you an example, when you take sample for real sampling for real patients, you often cannot go through multiple extractions. So what you gain is what you need to use to make an assessment. So definitely the sequencing is in the lower range, but and if you look at the number of CPG islands that you can detect, there's a huge difference, because so far you have a little less than 1 million CPG islands on the arrays, while when you do it with the reduced representation bisulfite sequencing, you have a range moving from the 2.5 million up to the 4 million. As I said, tools, including also the automatization, but you have already seen, for example, in the Margherita and Valentina presentation, this technology that we do have in the lab. If you remember the Oxford nanopore, and why I point it out here, because it's been introduced. It use directly the original nucleic acid and detect the base, also the modified base, just through the change on the current when we make the molecule goes through the pore. This has been one of the technology that we really push toward the COVID period. And the reason is that while we do have a population study machine that includes hundreds of pore, there's a lot of more feasible, much more smaller machine that still use the same kind of technology, still use the same chemistry, and that can be really used in simpler environment, or they can even use on the field. And this is why we do have in the lab the really high throughput machine that is the Promethean, but during the time we used many of the small one, starting from the really small one, the mini ion, the MK1C that many labs nowadays include for viruses analysis, and I'll show some data after. And finally, also the this new device to have thousands of pores, but with just few slides. And linking to the epigenetic through bisulfate conversion and through methylation study, this is a really nice example. Through the same sequencing with Oxford nanopore, you can see you can have a detection of the methylated cytosine directly without doing the bisulfate sequencing, so you don't go indirectly through mutation detection, but you have a direct detection of the methylation itself. So you can say that you can take two things with a pod, just making a single run. And what is more important here is that you can also include a different methylation state, that is hydroxymethylation of the cytosine. Where and how we use this kind of technology. During the time we built our collaboration around the concept of One Health. So it means that we use it for human health, but also for animal and environmental health. You can see some application has been made on genetics, on prenatal testing for cattle, for crops. We have done during the time lots of collaboration with the ICGB, the local hospitals, and also the veterinary institute in the region. But obviously, where we started aiming, and it was one of the reasons for the PRP project, was try to tackle the problem of the SARS-CoV-2 viruses. And this is a really nice example, because when they came in the lab and asked, do you think that it could be possible to sequence this kind of RNA virus? Nobody have actually, only in China they have done it. Do you think that from this sample from the patient it can be easily done? We quickly take what we already have done in the lab, we adapt the protocol, and we obtain the full length sequence of the SARS-CoV-2 in just a week, or a little bit less. And and that was quite fun, because it is actually the same protocol that you still use for the single cell for detecting the also not only the SARS-CoV-2 virus, but also the other transcript. And it was really useful to help to track at the really beginning the virus, and it brings also some kind of story, that because we, in that case, we didn't use the U1 sequence to recreate the sequence of SARS-CoV-2, but we make it available through the GSAID website. This is something that we built during the time through the hospital. So from the first sequence that you see here, we started uploading hundreds of sequencing during the COVID period. And while the protocol that I just showed you was a quick adaptation of what we already have in the hospital, through these many sequences, it was possible to build a target protocol that is basically, thanks to the fact that you share your sequence, the other can build on that. And for example, this is the basic how do you make a lot of detection of the virus in many different fields. In the sewage, for example, you can find it in small particulate. It was possible just because all the scientists that did that shared the data, and this was one of the great idea of building this lab around the concept of sharing the data, making the data available. This was really fun about the COVID period, because obviously sharing the data is also showed the needs during the time to keep sequencing, because you can detect the virus in many ways, much cheaper ways. But if you want to see if there's any variation, if there's any changes in the virus, the whole genome sequencing of the virus it's something that you need to do. This is a classical example. When we started sequencing the variants, it was pretty easy, I had to admit, because at the really beginning, the number of variants that you have in the virus were really bunch of less than 10 at the beginning until Omicron arrived and the situation completely changed. And as you will know, this is the reason why many of the approaches changed during the time. So starting from that, and thanks to a more target approach, it was possible to make to help a lots of different teams. So through the collaboration with the ICGB, we sequenced the virus for the Iraq, the Ethiopian group, Moldavia, Dominican Republic, but also we entered through the hospital on the weekly assessment of the virus during the SARS-CoV-2 period, and we started to realize it was a border. It's actually a good example of what can happen through the PRP. Once you achieve a technology that is stable, I think that is obviously no longer needed to be in center like ours. We need to spin out toward the hospital, toward the other groups that need in daily basis. And this is why we obviously this is something that I want to remember, because sharing the data made also possible the genomic surveillance and the statistic of the diffusion of the virus among the world, including in Africa. And we were really happy to participate also for the courses with the ICGB to teach PIs and students how to use this kind of technology. And I want to point out that it is just an example for a specific virus, but we have done for many other viruses. We have done for flu, we have done for monkeypox, and this is an example. So not only RNA-based, but also DNA-based viruses. As I was saying, we spin out this way how to detect the variants, actually to how sequence the virus in the hospital. And this was exactly why I showed you before the smaller instrument. This is the instrument that now the local hospital acquired, and how is now doing all the genomic surveillance on monthly basis. The COVID is no longer so of such interest anymore, but nevertheless we shared knowledge, and that was quite interesting because obviously the process, as I said, is well structured, is easy to deliver the kind of question that the hospital wants, that the people that want to react against the virus wants, at least in the term of knowing the variation, the genetic variation, and so knowing the amino acid substitution. But this is not something that we apply only for virus. We apply also for bacteria. And among the many ways how you can investigate bacteria, I just showed 3 of them. It's just a recent example, because you can take just a gene from the bacteria, is a particular gene, and through the sequencing of that specific gene, infer the microbial composition, or isolate the organism, do the whole genome sequencing, or try to pushing and through brute force sequencing everything, including all the things that are available on the sample. Obviously, this is the easiest way. You go through a specific gene, but since it's extremely variable among different bacteria, is somehow a proxy and easy. Is a marker to be used to detect at least the different kind of bacteria that you are involved with, still using knowledge and data that has been shared by scientists. But the real point here, obviously, this is because someone showed a multidimensional scaling last time about how they did it, the same things for bacteria, and we did the same things exactly with this kind of protocol. It's called 16S from the gene. And you see the multidimensional scaling, how the clusters the groups of things. Don't worry, this is not a new bacteria. This is a specific some publication that we have done on bacteria in cows and how they affect. You'll be really surprised that if you go on the Ministry of Health in Italy and you look for antibiotic resistance, they often coming from vet studies, and there can be really a big problem on the things how they proceed. But when we do the whole genome sequencing instead, after that we isolate the virus, the beauty of that is that you have all the information for the virus. It means for the bacteria, I beg your pardon. It means that you can also estimate even before you have tested for the antibiotic resistance if the bacteria itself is indeed one with the antibiotic resistance, what kind of antibiotic resistance is there. So in the first example, I just know the bacteria. In the second example, I know not only the bacteria, but how it might behave. Obviously, you need something else, but it's still a good starting point. And you obviously don't need to limitate yourself to just one of these. So when we integrate data, for example, for this detection of this specific bacteria, they started with Malditov. They detect the bacteria, and they ask. This is a collaboration that we have done through the hospital. This is actually a thesis of a student in biotechnology that is actually discussed the student this morning, if I'm not wrong. But the topic is really nice because they started with the hospital that was worried because many patients show the same characteristics, and they want to know if they suffer from the same kind of bacterial infection. And the easiest way to do so is 16S approach. But after that, we have done the 16S. We want to know how problematic is the situation. So we went to go through the sequencing of the whole genome, and we realized that indeed there's antibiotic resistance. And then we start going back where these bacteria entered the hospital, and we actually more they than us track back to drugs. So we use drugs that was contaminated with bacteria. And this is actually, you see there, it's actually been submitted to for a paper last Friday. But this is a perfect example how this kind of technology are tools that you can use to achieve something that can be used fighting pathogens, or at least helping the physician to have something to know how to what they're dealing of. This is a casebook study because the Malditov, the 16S, and the whole genome, they all point out toward the same things. But it's still a good example how we hope we can manage to use also the new technology for this kind of I need to rush a little bit, so I want to start closing, just pointing out that what I've shown so far is what we already have in the lab. It's not what the PRP is going to bring in the lab. And through the PRP, we're going to go through a second upgrades of the lab. So we are moving toward single cell analysis, spatial transcriptomics, and including the mixing of the data from different omics, especially try to entangle the proteomics, lipidomics, metabolomics through the classical genomics approach. And this is our challenge for the next year and a half, more or less, if I understand correctly. But the nice part that we did start already, even before the PRP was granted because we did start using the single cell. And this is an example how we traced back the COVID through the single cell in specific cells of the patients. This is an example of long COVID study. And not only the single cell is something that we started doing, but we also started to integrate for different topic, obviously, different omics. This is an example how we integrate data from single cell, bulk RNA-seq, metabolomic, and proteomics through this really nice collaboration that we have done last year. It is something definitely that we are still trying to do in many projects. Also, to give you an example, to integrate the transcriptomics and the methylation. This is the workflow that we use for this kind of integration. I don't think that I have enough time to go into detail of that, so I'll try just to go and closing, thanks to the people for the lab. I apologize. The slide is old. The lab is still hiring. For example, starting from September, Valentina will start working. I hope 100 of their times in our lab. So she's going to take charge of the cell expansion that we want to have, so the cell culture that we miss and we need to have. And I want also to add two more things before closing. First of all, that we are also strengthening our relation with the platform for proteomics in Salerno, and this will bring us that kind of integration that we need to move toward one single omics to multiple omics. And obviously this means that we are also hiring new people for that new facility, and this is something that we are going to do soon. So if you have some colleagues that are interesting, let's have a look, because we are going to put the tender actually, I think, in less than a couple of weeks. Thank you.
Thank you very much, Danilo, for your presentation. So we have had the chance to see what you're performing in your laboratory and are going to perform in the next months once everything has been set up within PRP at CERIC. Do we have a question for Danilo?
Thank you, Danilo. Very, very nice. Just a curiosity. I mean, use a single cell genomics, for example, to study cancer. There is sometimes the need to have access to DNA single cell information to study clonality within an organoid or a tumor or upon a treatment. So do you have in place the technology to run this type of experiments or not? Because so far, for example, the 10X is not providing anymore this type of let's say, kits and technology.
Yeah, I have to admit that I'm really bluffed that the next start ruling out many protocols that are still used, and the one of DNA is one of these. The truth is that there's a lots of new technology that do not require some kind of instrumentation. Just to give you a short answer, not yet. We are working on it. It is actually something strange, because if you think about 5 years ago, it was really available in the market and you can do it. But if you want to do it right now, you must rely on some kind of specific homemade protocol and make the things a little bit more difficult, because obviously whenever we can, we try to rely on something that is stable, that can provide the user something that they can really trust. But you pointed a weak spot, actually, because it's something that surprised that this kind of protocol has been cancelled.